Okay, so the last thing I want to talk about today is life at high temperatures. There's a lot of interest in life at high temperatures, a lot of utility for the organisms and their enzymes that grow at high temperatures can be put to. There's also a lot of interest in them from the space program because there is good reason to believe that life may have originated at high temperatures and that life may exist in other places than Earth, in places that are high temperature, things like hydrothermal vents on Europa, etc. None of that's been confirmed, and there actually isn't any good evidence for it, but it is conceptually possible. There's actually more interest from NASA these days for life at low temperatures. So there's a lot of research going on in Antarctica and these permanently frozen over lakes in Antarctica as model systems for life in places like Europa that have an entirely ice crust on the surface and a liquid water ocean underneath. There's some really cool experiments that have to do with that NASA's proposed. Probably aren't going to get funded, but still would be interesting. But high temperatures is really interesting. And so this is Obsidian Pool. What are some of the issues that organisms have to deal with? I mean, if we took you and put you in a pool like this, you would die quickly. What goes on? What is required of an organism to live at these temperatures? Well, one of them is membrane fluidity, right? You've got, all your cells have membranes, and those membranes are two-dimensional fluids. The proteins in them can float around unless they're tethered. The actual lipid molecules within them diffuse through that two-dimensional lattice. And that membrane fluidity is critical for the function of the membrane. As you increase the temperature, the diffusion rates in that membrane increase, the lipids are jiggling around, and ultimately the membrane is disrupted. And so in order to function at high temperature, you have to change the lipids in your membrane, make them less fluid at any particular temperature, so that at the temperature the organism lives at, they have that correct fluidity. This turns out to be really easy. It's straightforward to decrease things like unsaturations in the lipids, to decrease the number of side chains. There's a variety of things you can do to the lipids to make them have the right fluidity at a higher temperature. In some cases in archaea, you actually have lipids that span the entire cross-section of the membrane that kind of tether both sides of the membrane together. Bacteria grown at different temperatures tune their lipids to grow for optimal function at that temperature. So E. coli growing at 30 versus 37 have different membrane composition. So this turns out is probably pretty easy. DNA structure sounds like a terrible issue, right? I mean, if you take DNA in water in a more or less aqueous solution, it probably has, say, 5 millimolar magnesium. It'll denature at about 65 degrees. That is, the two strands will come apart because the hydrogen bonds are strong enough to resist that thermal vibration. 65, 70 degrees. And that depends a little bit on the base pair composition. GC pairs can strengthen it some. If the purines are in a row, they stack on top of each other, and that increases their thermal stability. But realistically, DNAs all melt at about that temperature range. So how does an organism grow at 95 degrees if its DNA wants to melt at 70? That turns out to be easy. Proteins bind to it to stabilize it. Many thermophilic organisms will wrap their DNA around proteins to help stabilize them. We call those histones in eukaryotes. We call them histone-like proteins in bacteria and archaea, which probably evolved for this purpose and not for packaging. It also turns out that the aqueous environment where you take this kind of a measurement in has nothing to do with what DNA sees in the cytoplasm. That thermal denaturation temperature depends fundamentally on the ionic conditions and on molecular crowding. And DNA in mesophiles is negatively supercoiled specifically to make it possible to separate the strands of DNA during transcription and during replication. And so these organisms don't negatively supercoil their DNA as much. And in fact, some thermophiles have a reverse helicase that positively supercoils, cranks the helices tighter to make them more resistant to denaturation. This turns out then to be easy. By taking these sort of things, you can crank the DNA melting temperature up well beyond 100 degrees. RNA structure. If you take a ribosomal RNA, for example, and put it in a tube of water and heat it up, it quickly denatures. The tertiary structure comes apart first, and then the secondary structure, all the base pairs come apart. Like protein, this is usually readily dealt with. Some of that is taken care of by changing the sequence and structure of the RNA. And so if you take, for example, a ribosomal RNA from a thermophile and a mesophile, you find out that the thermophilic ribosomal RNA has a higher melting point than the mesophilic one. And so that's an inherent property of the sequence and structure of that thermophilic RNA. But these things all bind proteins. They're crowded together. There are lots and lots of things that the organism can do to make them resistant to denaturation. In the case of the RNA itself, I mentioned the sequence. You can also decrease the irregularity, shorten the length of the RNA. This is one that people forget about. If you've got an RNA with all the extras taken out and is shorter, it will be more thermal stable, because thermal stability has to do with binding energy and alternative foldings. And the shorter the RNA is, the fewer the alternative foldings. It makes it more resistant. You can put extra GC pairs in there, but you can only go so far with that. You can modify bases. You can do things like put thiol groups in. You can put methyl groups on. There are lots and lots of things that you can do to an RNA to make it thermal stable. And organisms do that. Protein structure is a really interesting one. An expert can look at an RNA, a thermophilic RNA and a mesophilic RNA, and can tell you which is which, just by looking at the idiosyncrasies of the RNA structure. Not so proteins. To this day, a biochemist can't look at, for example, glyceraldehyde phosphate dehydrogenase and look at them side by side and tell you which one will function at high temperatures. That's pretty remarkable. And if you denature proteins, that's how you make eggs, right? Put them in a frying pan and then proteins denature. It's pretty dramatic. What's happening? What's happening is the outer hydrophilic layer of the protein is being so destabilized that the hydrophobic core opens up to the environment. And adjacent proteins will latch onto that, and the hydrophobic force will force them together. And the result is you get this giant network of hydrophobic strands, and we call that denatured protein. How do you stabilize a protein against that? It turns out that there are some forces that hold a protein together that weaken with higher temperatures. Electrostatic bonds, hydrogen bonds, and ionic bonds get weaker as the temperature goes up. Keep in mind that thermal vibration is also fighting against them, right? There's more jiggling going on in the protein. But hydrophobic forces get stronger at higher temperature. If you're really a biophysicist, you'll say that I'm simplifying it too much. What's actually happening is that the hydrogen bonds in the water surrounding the protein are getting weaker. The hydrophobic force is the absence of solvent hydrogen bonding, right? And so what you see in thermophilic proteins is stronger hydrophobic cores and less reliance on things like ionic and hydrogen bonding in the outside. And how organisms do this, how these proteins do this, is a bit of a mystery. But the reality is, if you take a protein from a thermophile and measure its thermophilicity, you'll find that it's a lot more thermal stable than that of a mesophile. And this is absolutely critical, particularly for enzymes. Enzymes aren't machines in the way we think of them. They're not metallic objects that have specific gears and things. They're pretty squishy. They're more like jello than metal. And they have to bend. That's part of their function. They have to be dynamic. They have to open up that cleft in the active site so the stuff that is binding to it can come in, and then it has to change shape during catalysis. That dynamic flexibility is required for its function. And so proteins are evolved to have the right amount of flexibility at the temperature they function at. At much higher temperatures, they denature, and at much lower temperatures, their catalytic activity goes away because they can't move. And so these proteins evolve to match the temperature that they need to function at. And it seems, although we don't understand it, it seems to work pretty well. Enzymatic function is a big one. So if you take an entire pathway and shift it to a higher temperature, that pathway will go haywire. Because previously, all the rates were titrated so that things would progress one to the next and things would siphon into the right places. The rates and activities are tuned for that temperature. As soon as you change that temperature, those rates all change relative to each other. Some reactions go faster, some reactions go slower, and the whole pathway goes haywire. And so you have to tune the amount of catalytic activity to drive reactions the way you want to go. Think about it this way. If you've got a unimolecular reaction, something that binds a substrate, makes a change, and throws away the product, it will get more active at higher temperatures because diffusion is usually rate limited. It has to bind the substrate. And as that diffusion rate increases with higher temperature, it will go up more or less linearly as the temperature goes up. But what if you have to bind two substrates? Now the diffusion rate to get both of them loaded into the enzyme goes up with the square of the temperature because you're binding two substrates, each of which has to diffuse in. So those enzymes, the rates will go up much more quickly. And you're likely to start pushing the accumulation of intermediates, and things get siphoned off into other processes, and you have a nightmare. And so organisms have to have homeostasis. They have to be able to adjust enzyme activities in various ways to keep them functioning the way they need to. But we know how to do that, right? You change gene expression. You have regulatory mechanisms that control the production of enzymes or the activity of enzymes to suit your needs. So that turns out to be pretty easy. And this is just showing you, I showed you that already. That is, that thermophilic enzymes work at high temperatures, organisms or enzymes from cryophiles work best at low temperatures, etc. Small molecule stability is a big one. How many of you guys work in a lab that does molecular biology? Where are the nucleotide triphosphates? In the freezer, right? Yep, typically minus 80, sometimes in the minus 20. Why is that? Because they're not stable, right? Now, ATP is pretty stable, it turns out. And you can have a solution of ATP and leave it out on the bench and it's just fine. Not so GTP. GTP hydrolyzes spontaneously at a pretty significant rate in the presence of any measurable metal ions. And so GTP has a half-life of 3 seconds at 100 degrees. How does an organism live at 100 degrees? You need GTP for everything, right? For translation, for the Krebs cycle, et cetera, et cetera. How do you deal with that if you're a thermophile? And it's not just GTP, but it's things like UTP and NAD and so forth and so on. Many of these small molecules aren't very stable and will hydrolyze at high temperatures. Probably what these organisms are doing, one is they stabilize them by molecular crowding. You just don't have a lot of available water in the cytoplasm. Oftentimes you'll have compounds in the cytoplasm that are thermal stabilizing. But mostly it's a matter of producing it just in time. It may be that the half-life of GTP is 3 seconds, but it may also be that in the cytoplasm, GTP is on average used within 100 milliseconds of its production, in which case the hydrolysis rate isn't really very important. Yeah. So by producing these just in time, you eliminate this problem. And the reality is organisms seem to be able to do that pretty well. Intermediates and biosynthetic pathways may have a short half-life, but they're handed directly from enzyme to enzyme. And so their flux is very high, but their time in existence is very small. Right now, the best contender for what actually limits how high in temperature organisms can live is about chirality. If you had, yeah.
So I know you have 140 degrees Celsius. Is the pressure different for the
Yes.
water and changes?
Right. So these are typically underwater environments, and so the hydrostatic pressure increases the boiling point of water. You only have to go about 100 feet underwater before the boiling point of water is in this temperature range. So what happens is L amino acids get flipped to D. You've got these bonds, right? But those bonds are also subject to thermal vibration. And at some rate, a chiral center will have, I can't do this with my fingers, will have one of the bonds flip over and invert. And now you've changed a D amino acid to an L or an L to a D. You've changed a D sugar to an L sugar. That's a disaster, right? These things are typically pretty toxic because they inhibit enzymes. Imagine what effect the flipping of an amino acid in the middle of a protein has. It typically will cause denaturation of that protein. And that inversion or racemization of amino acids has pretty significant rates at temperatures of 130 to 150 degrees centigrade. And may represent then the kind of ultimate limit for how high a temperature you can live at. I say may because although people generally agree this is a great big problem, it wasn't so long ago that nobody thought that things could live at 90 degrees because of the half-life of GTP or because of DNA denaturation. So every time someone has proposed one of these ultimate limits, it's been found that organisms have a way around it and can grow at higher temperatures. There are reports of life as high as 350 degrees, almost surely not true. The best solid evidence is for life at about 140 degrees. If you take microscope slides and put them in deep sea hydrothermal vents at 140 degrees, things grow on them. You can't cultivate any of those creatures, but they seem to be living and growing at that temperature. Anything higher than that, and there's no evidence for it. But that doesn't mean it isn't real, right? Can you think of any ways that organisms might be able to get around this problem? I'll bet you can. No evidence for an enzyme that can do that inversion, by the way. That would take a ton of energy. All right. So with that, I'm going to let it go.